Hello everyone, my name is Gary O'Mealy and I'm an assistant professor of biology at Tulsa Community College, and this is the first video in my new video series on human physiology. This video pertains to homeostasis. Before watching this video, you should have hopefully read sections 1.4 and 1.5 in the OpenStax textbook. Let's start by defining very quickly what homeostasis is and why there is a need for it. The first thing to understand is that the body maintains steady internal conditions despite the very natural tendency of those conditions to either change over time or in response to changes in the external or internal environment. A good example of this is that without an air conditioner, your house's temperature would change to match exactly what the temperature is outside. Is it hot outside? Well, the inside of your house would be hot. Is it cold outside? Well, the inside of your house would be cold. So having an air conditioner or a heater helps to make sure that the inside of your house is very comfortable despite how the temperature outside has a very natural tendency to change. And what I'm trying to convince you of here is that your body operates very much the same way. Now, in order to actually coordinate homeostasis responses in the body, this is going to require the constant monitoring of internal conditions by what are called sensors. Now, your sensors are going to be monitoring all sorts of things, including your body temperature, your blood pressure, the oxygen levels in your blood, the availability of glucose and other nutrients, and the list goes on and on. And as we go throughout the class this semester, we will introduce a myriad of other examples of this as well. So the idea here is that your body's function will greatly suffer if any of these conditions rise or fall to undesirable levels. The common theme here is that we don't want any of these things to get too high or too low, and homeostasis helps us to prevent those sorts of things from happening. So, as to what a sensor is a cell or group of cells in the body that is sensitive to changes in a particular condition. And what we're going to see here is that sensors tend to be very particular about what they are sensing. You can see a variety of different kinds of sensors here, and you can look at what they're detecting and relate them to just about any kind of feeling you've ever experienced yourself. Mechanoreceptors are responsible for your sensation of touch and temperature and the fact that you can hear. Chemoreceptors allow us to taste and smell, and then internal chemoreceptors help to allow the body to detect how much oxygen is in the body, how much carbon dioxide is in the body. Photoreceptors help us to see. Baroreceptors help us to maintain things like blood pressure. Osmoreceptors are responsible for detecting when water levels are out of kilter and are responsible for things like when you feel thirsty. And nociceptors are responsible for your sensation of pain, which, undesirable as it may be, is extremely important. Now, an important thing to understand about the limitation of sensors is that they lack the ability to do anything other than detect when conditions change and then pass the message along. It's helpful to think of homeostasis as being something like problem solving. When a problem comes about, homeostasis helps to fix it. Well, we need to understand that sensors are not there to fix the problems themselves. Sensors can only notice that there is a problem to begin with and then pass the message along to someone who is more capable than themselves. So what's going to be necessary here is efficient and faithful cell-to-cell communication in the body. The sensor has to be able to communicate the nature and severity of the problem to cells elsewhere in the body, and those cells are going to be the ones that help to coordinate the response that will fix the problem. This faithful cell-to-cell communication is going to be absolutely essential for any homeostasis mechanism to do its job to solve problems. Now, you can sort of think about each part of a homeostasis loop looking like this. The stimulus represents whatever the problem is. High blood pressure, low oxygen levels, high blood glucose, low body temperature. The stimulus is going to be numerically some internal condition being too high or too low. The sensor is going to be that group of cells that is actually strategically positioned close to where the stimulus is at its most intense so that the sensor can be well positioned to detect when the stimulus becomes a problem. Well, like we said, the sensor can't really do anything other than notice that the problem is there, so the sensor is going to have some way of communicating the nature and intensity of that stimulus to some other part of the body. This line of communication that the sensor sends out is called an afferent signal, or sometimes called an input signal. And you'll notice here that this signal is being sent to another group of cells in the body called a control center, which we will detail in just a minute. So the sensor will communicate the status of the conditions to a control center, and typically in most cases when you're talking about a sensor communicating to a control center, this message is carried by either a cranial or a spinal nerve. A cranial nerve carries messages into and out of the brain directly, and a spinal nerve carries messages into and out of the spinal cord. So we're going to see examples in the future of, in some cases, the sensor and the control center are one and the same. Some sensors are actually embedded within the brain itself. Now, a lot of these sensors make up part of what we call sensory neurons. So if we look at the anatomy of a neuron, which you'll learn more about when we actually start properly covering the nervous system, the neuron has dendrites, a cell body which contains the nucleus and other typical organelles you would expect a cell to have, and then an axon for passing the message along. Now, it's the dendrites that's going to be the actual location of the sensory receptors, which are proteins that actually allow this sensory cell here to detect the presence and intensity of that stimulus. Now, if the stimulus is intense enough, those sensory receptors will be activated and a message will be transmitted along the axon of this sensory neuron, which is called an action potential. And those action potentials, which are electrical in nature, will be sent from this sensory neuron, which is positioned close to where the stimulus is, all the way to wherever the control center is, whether that's the brain or the spinal cord. The brain and spinal cord are likely a long way away from wherever this stimulus is, so the sensory neuron here helps to detect the stimulus and then get that message very quickly delivered to some part of the body that will be able to coordinate some sort of battle plan for how are we going to address this stimulus, whatever it may be. So when you think of something like a spinal or a cranial nerve, a big part of those nerves tend to be made up of the axons of these sensory neurons. So we can send a lot of messages back and forth between different parts of the body at any given time. Now, if we look at the back end of a homeostasis loop, the control center, of course, receives that incoming sensory information and works to formulate an appropriate countermeasure to restore homeostasis. If the blood pressure is too high, we got to figure out a way to bring it back down. If the blood glucose is too low, we got to figure out a way to bring it up. The control center is made of cells, usually in the brain or spinal cord, but sometimes in an endocrine gland, that are capable of determining what the best response is to a particular stimulus. The sensor just does not have that processing power, so that's why it's important that the sensor can communicate with the control center. Now, the control center, once it has figured out a good strategy of dealing with the stimulus, it will then send its own message, which is called an efferent message or an output signal, from the cells in the control center towards cells elsewhere in the body, which are called effectors. Now, these messages that are sent from the control center to the effector don't necessarily have to be electrical in nature. Now, certainly they can be and often are, but sometimes these messages, instead of being sent through nerves, can also be sent as blood-borne hormones. So if you think about the way that long-distance communication works in the body, we've already seen one example of this. In the case of a neuron, which you can see up here, in this case a motor neuron, which is going to send one of those efferent output signals that we saw before, it's going to generate action potentials which carry down the length of the axon, And then once the action potential reaches the end of the line here, the end of the axon, which is called the axon terminal, will release a chemical called a neurotransmitter. And that neurotransmitter is going to carry the nature of the message that the control center is trying to send to the effector cell. But like I said, that's not the only way this can occur. An endocrine gland, which you can see down here, can secrete a hormone, which should be fairly similar to the neurotransmitter in terms of its chemical structure, but instead of this message traveling through a long nerve, this hormone travels through the bloodstream, and then eventually the hormone exits the bloodstream and then binds to that effector cell, essentially delivering a message, albeit in a slightly slower fashion, but still an effective one nonetheless. Now, the common thread I want you to notice here is that both of these are forms of long-distance communication, and despite their differences, both forms of long-distance communication, whether it's a neural message or a hormonal message, involve a chemical signal binding to receptor proteins on the effector cell. That is absolutely essential to producing the response to the initial stimulus. You have to have a chemical signaling molecule, whether it's a neurotransmitter or a hormone, activating a receptor on the effector cell, and that interaction between a neurotransmitter or hormone and its receptor is going to produce a change in effector cell behavior that is eventually going to lead to a response which should hopefully counter whatever the initial stimulus was. And if that seems kind of vague for right now, that's okay, because we are going to see a lot of examples of that as we go throughout the course. So the effector, in receiving this efferent message from the control center, whether it's a neurotransmitter or a hormone, this is going to produce a physiological response that will then cause the initial stimulus to diminish. So that's very important. The response here should be the exact opposite of whatever the stimulus was. If the stimulus was high blood pressure, the response should be something that lowers the blood pressure. If the stimulus was low blood glucose, the response here should be something that raises blood glucose. So this type of homeostasis is what we call negative feedback, which we will more aptly define here in just a minute. If we follow the entire loop around, if we say the stimulus is high blood pressure, the sensor detects the high blood pressure, sends a message to the control center, let's say the brain, to tell the brain about the high blood pressure. The brain then communicates, let's say, with the heart, saying, hey, the blood pressure is high, you need to kind of slow your heart rate down so the pressure doesn't get too high. The heart will receive a message from the brain. The heart muscle cells will respond by beating less quickly, and that will cause the blood pressure to start going down. Now, what makes this loop shut off, which is why we call this negative feedback, is because there is basically a halting of the cell-to-cell communication going on here. So the idea here is that in negative feedback, the induced response reverses the course of the sensed stimulus until it completely goes away. Okay? So if the response here is that the blood pressure goes down, eventually we're going to bring the high blood pressure back down to normal, to the point where the sensor no longer has a stimulus to sense. If the blood pressure is normal, then there is no stimulus. So basically the way to think about this is that the removal of the stimulus causes all lines of communication in the loop to cease, meaning the whole response is going to shut off automatically. Think about it this way. If the blood pressure goes back to normal, the sensor no longer has a stimulus to sense, so it's going to stop its communication with the control center. If the control center is no longer receiving communication from the sensor, it will no longer send messages to the effector. And if the effector is no longer receiving messages from the control center, it will stop doing whatever it was that was bringing the blood pressure down to begin with. And we're going to see that this is very important because this prevents us from overshooting our response. If the blood pressure is high, we don't want to bring the blood pressure down so much that eventually we create a new problem for ourselves by making the blood pressure too low. So basically, this is going to lead into kind of the next question. What exactly qualifies as a stimulus? So the idea here is that the body doesn't really need to waste energy on trying to fix every little change that occurs. If the blood pressure goes down by 1 millimeter mercury, or if your body temperature goes up by 0.1 degrees, that doesn't necessarily qualify as some major wholesale change that the body needs to freak out about. The idea here is that it's only large deviations that require engagement of homeostasis loops. Things really need to go wrong for the body to actually respond in a certain way. So that's going to lead to us defining a couple of new terms here. So every measurable parameter in the body has what we could consider a desired set point. So if you think of something like body temperature, you're probably aware that your average body temperature is about 98.6 degrees Fahrenheit or 37 degrees Celsius. That is not exactly what the body temperature has to be every single second of every single day. It's not the end of the world if your body temperature is 98.0 or 98.9. Not the end of the world. In fact, that is very, very, very normal. It's okay if the parameter is not at the set point all the time. So let's look at a graph that will actually illustrate this. And since I brought up body temperature, let's use that as an example. So let's define what our set point is here. We'll say set point is 37 degrees Celsius, as I said before. Now, the way homeostasis is going to work here is that we establish an upper limit for which that is our limit in terms of we do not want the body temperature to get any higher than this, and then a lower limit. We don't want the body temperature to get any lower than that. So the idea here is that if you were to measure your body temperature every hour for a whole day, each measurement is not going to be exactly the same. Some measurements may be right at the set point of 37 degrees. Others are going to be a little higher and others are going to be a little lower. So if we actually look at what the data would look like, it's probably going to look a little something like this. The body temperature, just like any other homeostatic parameter that can be measured, like blood pressure, blood glucose, and things of that nature, there's going to be fluctuations. And the fact that there are fluctuations, but that we don't ever allow the parameter to get above the upper limit or below the lower limit, this is evidence that homeostasis is working. When the body temperature gets too high, we sweat to bring it back down. When the body temperature gets too low, we shiver to bring it back up. And that ignores less obvious manners of thermoregulation, like the constant production of heat from your metabolic activities, the nature of that heat to escape from your body, but these are just kind of more specific examples for homeostasis. The main takeaway here is that all your different homeostatic parameters like body temperature and blood pressure are going to fluctuate, but as long as they don't get too high or too low, that's just evidence that your homeostasis is doing its job. So the idea here, if we want to sum this all up, is that as long as the parameter is within the normal range, everything's okay. There's no problem as long as you're somewhere within that normal range, either a little bit above the set point or a little bit below the set point. If you're in that normal range, you're all right. So the set point is just the desired value of the parameter on average. It doesn't mean that parameter has to be the set point every single second of every single day. So if you consider homeostatic mechanisms like sweating and shivering, these don't get activated until the limits of that normal range start to get breached. So in doing this, we save the body a tremendous amount of energy by determining that this is how much a parameter has to change before we say enough is enough and we need to step in and fix this. Now, if you want some more examples of different parameters and their normal ranges, you can see blood glucose has a normal range of 70 to 110 milligrams per deciliter. Systolic blood pressure has a normal range of 90 to 115 millimeters mercury. The pH of the blood, 7.35 to 7.45. Blood osmolarity, 285 to 305 milliosmolar. So you can kind of see what these normal ranges look like, but if you take a peek at the far right column, which is listed under the header disease, you can see examples of diseases that are associated with these parameters being outside of the normal range. Diabetes mellitus is characterized by high blood glucose. Hypertension is characterized by high blood pressure. Acidosis is characterized by low blood pH. And dehydration, in addition to things like diabetes insipidus, are characterized by getting outside the normal range for blood osmolarity. So hopefully this is kind of getting you to think about normal human physiology and the nature of disease. We can very quickly and very easily sum this up, that disease is a reflection of the body's failure to maintain homeostasis. So if you consider something like diabetes at the cellular and molecular level is a failure to properly regulate blood glucose. So that's why in this class it's going to be very important for us to become very closely familiarized with these cellular and molecular mechanisms so that when a patient does come in, say, with high blood glucose, you're going to know exactly where to look and have a good idea of exactly what is wrong with them at the cellular and molecular level. Now, of course, if we're going to talk about negative feedback, that obviously implies the existence of positive feedback too. Now, in most cases, positive feedback is not something that would be all that useful to us, because what positive feedback implies for us is that it would amplify the intensity of that initial stimulus rather than reducing it like negative feedback does. Very much like a snowball rolling downhill that picks up more and more mass and gains more and more momentum. So if we look at the same sort of graph that we looked at before, except for positive feedback instead, a positive feedback mechanism, as soon as you breach that upper limit, would actually accelerate the trajectory of that parameter to make it even more out of the normal range than it already was. Now, hopefully you can appreciate that that would not be something that we would want to happen in most cases. If our body temperature was regulated by positive feedback, anytime we start sweating, our body would get hotter and hotter and hotter instead of cooler, like we want. So in most cases, positive feedback would just make the problem even worse than it was before. But we're going to find in this class that there are very specific situations in which positive feedback is actually a good thing that actually accomplishes something very useful for us. So a good example of this is in childbirth. The idea of childbirth is that once a baby reaches the end of the gestation period, it starts to descend down into the birth canal, and in doing so, it starts to place pressure on the cervix. This increase in pressure from the cervix causes an input signal to be sent from the cervix up to the brain, and the brain will then stimulate the secretion of a hormone called oxytocin, which is carried from the blood back to the smooth muscle here in the uterus, and that causes the uterus to produce contractions, which pushes the baby even further down, pushing even more pressure against the cervix, and that's going to cause this snowball to start rolling and rolling and rolling and rolling. So the contractions cause there to be even more pressure on the cervix rather than less pressure, so that's going to create this positive feedback loop where we secrete even more oxytocin, and over time that makes the contractions even more intense and pushes the baby even further down. So obviously, if we graph this out compared to how we did before, the stimulus would be the increased pressure on the cervix. The cervix is the location of those sensory neurons, sensory receptors, which are connected via a sensory nerve that connects to the brain. The brain receives that message and stimulates the posterior pituitary to secrete oxytocin, which travels through the bloodstream towards the effector, which is the uterine smooth muscle. The smooth muscle produces contractions after being stimulated with that oxytocin, and rather than making the stimulus go away, those contractions actually cause there to be even more pressure on the cervix, and that's going to cause this part of the loop to repeat even more strongly than it did before. Now, the question that you might be asking is, well, what is going to actually ensure that this loop will eventually shut off? So go ahead and look at that diagram again of the baby being pushed even further and further down into the birth canal, and I think you'll come to the correct conclusion on your own. Okay, that's going to wrap it up for this video. Here is a list of vocabulary terms that I think you ought to at least be familiar with if you don't already have a good handle on them. And then here is a check your understanding list. The first question, why is long-distance communication important to homeostasis? What are the two methods of long-distance communication that the body uses to maintain homeostasis? Why do blood tests measure all sorts of parameters for patients? And finally, why is negative feedback much more common than positive feedback in the body? So that's going to wrap it up for this video. Hopefully you'll look forward to video number 2. See you next time, and have a good day.